class, what we have talked about was, we were talking about the development of endosperm. Yes or no?
Yes, ma'am.
Right? So in the last class, what we were talking about was, we were talking about the development of endosperm, and basically under which heading? Post-fertilization stages, right? So post-fertilization stages for the endosperm is the first step that we were talking about. What we have discussed is who forms endosperm, what is the body of endosperm, etc., etc. And the topic that we were discussing was the types of endosperm. No, depending on how they are formed, endosperm could be classified into three types: that is cellular, nuclear, and haloidal. In the last class, we have completed the discussion about nuclear endosperm. So today we are going to start the class with the next type of endosperm formation, that is cellular endosperm. So please make the heading cellular endosperm. So आज की class में types of endosperm. Continuation. So types of endosperm, nuclear we have done. We are going to do the number 2, that is cellular endosperm. So again in the form of a diagram, not in sentences. So since we are talking about endosperm, kuntu cell of the beginning of the cell that will be responsible for endosperm formation is called as Kibri. পাহৰিলো নেকি?
কি কোশ্চেন উঠিছে মেম?
এণ্ডোস্পাৰ্ম ডেভেলপমেণ্টৰ কোনটো চেলৰ পৰা বিগিনিং হ'ব প্ৰচেছটো?
Pen.
Yes, it's pen.
Primary endosperm nucellus. Yes ma'am.
Nucleus. Primary endosperm nucleus, PEN.
Nucleus na nucellus ma'am?
Nucleus.
Okay ma'am.
So no sentences, only the diagram. So when we talk about types of endosperm, last class of nuclear endosperm, and what we remember that in case of nuclear endosperm, what happens? The nucleus undergoes free nuclear division. No, free nuclear division. It's like the entire embryo sac is filled with, or it's filled with only nucleus. We have talked about the development of the vacuole, right? সেইবিলাক আমি কলো, and later as the cell gets mature, তাৰপিছত কি হয় cell formation দেখা পায়. But in this case, it's the normal cell division that we talk about, right? প্ৰথমতে কি হ'ব, the nucleus will divide into two. Cytokinesis will immediately be followed by cytokinesis, and the process will go on. Since we see cell formation at the beginning itself, so this type of endosperm formation is called as the cellular endosperm. Nuclear endosperm cell formation দেখা পাইছিলো নাই? Nuclear endosperm cell formation হৈছিলে নে নহৈছিলে? It was in the, yeah, it was in the later stages, right? The cell formation gradually started, but in this case, at the very beginning itself, the normal type of cell division. So the examples are important. We have already discussed about coconut milk. Now we are talking about coconut milk. So this is the second type. Next, we have third type. ঠিক আছে.
Example to coconut meal লিখিছে.
Meal, সেইটো coconut meal বুলি কয়, যিটো white portion আমি খাওঁ, that is called as coconut meal. Cellular endosperm হ'ল নেকি?
Yes, ma'am.
Ma'am, explain robot.
Okay.
I'm writing it in English.
Okay. এতিয়া মুচিব পাৰো নেকি তলৰখিনি?
Yes, ma'am.
Yes or no?
Yes, ma'am.
লিখিলো এইখিনি So this is the third type of endosperm development that we talk about, that is haloidal. Right? Another type of endosperm development is considered as an intermediate type. Intermediate মানে দুইটাৰে মাজৰ কোন দুটাৰ মাজৰ নিউক্লিয়াৰ আৰু সেলুলাৰৰ মাজৰ স্টেজ বুলি কৈছে কাৰণ নিউক্লিয়াৰত আমি কি দেখিছো যে নিউক্লিয়াৰ ডিভিজন দেখিছো সেলুলাৰত from the starting point itself কি হৈছিলে cell formation হৈছিলে whereas we see the both the process in case of haloidal endosperm and so it is considered as an intermediate type, right? Since this type of endosperm was found in the order haloidals of monocots, so this endosperm was named as haloidal endosperm. ইয়াতে আমি কি পাওঁ যে দুইটা type of endosperm দেখা পাওঁ এটা part কি form কৰিব cell formation কৰিব and they will form the endosperm. Which type of endosperm? Cellular endosperm. আৰু ওপৰৰ পিনৰ this cell ultimately pre-nuclear division হ'ব and the same process as we have discussed in nuclear endosperm. Is the point understood?
Yes, ma'am.
দুইটাৰে combination থাকিব ঠিক আছে so this was all about endosperm development. So we are talking about the post fertilization changes of events and the first event that we are speaking about is the endosperm formation. Endosperm formation কি কৈছিলো যে it is seen or it is a result of double fertilization and double fertilization is a unique characteristic of angiosperms. Endosperm formation is also seen in angiosperms. We have already talked about it, right? কেনেকে endosperm develop কৰিছে depending on that we have two types of endosperm and the examples accordingly. So this is all about the first one. Second, we have to talk about the next post-fertilization event. That is, we have to talk about now the embryo development. So fertilization of this sort, embryo development, endosperm development, seed formation, or fruit formation. So first point we have completed. Second, we will be talking about embryo development. And when we talk about embryo development, obviously Dicot embryo and Monocot embryo. You know, before going into embryonic studies or embryology, point আমি লিখি লব লাগিব এইখিনি হৈছে নেকি
Ma'am,
Yes, ma'am.
and cell wall laid between two nuclei মানে কি হয় ma'am?
cell wall laid between two nuclei মানে এটা cell আছে ঠিক আছে cell আছে এইটো নিউক্লিয়াস আছে normal cell division এইটো cell যেতিয়া cell divide কৰিব তেলে first nucleus দুটা divide হ'ব নহয় This is a normal cell. This normal cell will undergo cell division. Jadi cell division hobo lage. Cell division at first kinesis. So obviously the nucleus divided and form two nuclei. After that, followed by cytokinesis. So this is what is the meaning of that line. Cell wall laid between the two nuclei. Jadi cell wall is not laid between the two nuclei. Ekon jodi form nokorile hete. Nokori etu jodi akko eta form kore. Etu jodi akko eta form kore. Similarly, the number of nuclei keeps on increasing. Teতিয়াhole ki hobo? Nuclear type of development? Yes or no?
Yes, ma'am.
So is the line understood now?
Yes, ma'am. Thank you, ma'am.
Okay. Okay, welcome. হৈ গলে মোক কবা এটা ডায়াগ্রাম পোর্শন হয়েছে থাকে ন
Yes.
over লিখিব মুছিব পাৰো নে
Yes, sir.
certain points
Ma'am, bracket কি লিখিছে?
coccus lucy the scientific name of coconut
Okay, ma'am.
hmm.
Ma'am, Zeniatur, ma'am, the effect of foreign pollen on endosperm.
On
Sorry.
endosperm character.
Okay, ma'am.
On endosperm character.
Ma'am, maize endosperm of কি লিখিছে?
Uh-huh.
Maize endosperm of কি লিখিছে?
Color. Color. Endosperm হল no? Normally endosperm বোৰ triploid বা triploid, but we see diploid, double endosperm. Right? Next, talking about genia or metagenia, direct question ahe, basic details nalage, direct question kya hai, what is the effect of foreign pollen on endosperm called as, what is the effect of foreign pollen on somatic tissues called as, right? So ultimately ami ki sa on endosperm aru on somatic tissue, just two terms, basic details nalage, ketiaba ahibo pare, effect of genia was observed in, so it is observed in. Next. Right? Foreign pollen on endosperm means what? যে যোনটো pollen তাক fertilize কৰিব লাগিছিল বা double fertilization part ল'ব লাগিছিলে সি নলৈ পেলাই বেলেগ এটা pollen tube penetrate কৰি বেলেগ male gamete যদি endosperm formation কামটো কৰে, then that is what is called as effect of foreign pollen on endosperm. Is the point character? Sorry, is the point understood? ফৰেন পলিন বুলি কি বুলি কৈছো যে বেলেগ পলিন টিউব পেনিট্ৰেট কৰিব পাৰে সেইবিলাক বেলেগ পলিন টিউবত যিবিলাক বেলেগ আছে যে তাহাতে যদি they what have an effect যে because of that work no because of penetration of that foreign pollen tube তাহাতৰ কি হল এন্ডোস্পাৰ্মৰ কিবা অকমানৰ characters বিলাক change হল Another term that is mosaic endosperm, an endosperm with sugary and starchy parts forming patches of yellow and white. So this is called as a mosaic endosperm.
Ma'am, second example to কি হয়? Ruminate endosperm.
কিহৰ? Annona. AMMONA.
ANN ne AMM?
ANN.
Okay, ma'am.
So initial points. After these points, we have to talk about the next stage, that is embryo development. লিখি লোৱা হলে মোক কবা। হ'ল নেকি?
Yes, ma'am.
চবৰে হ'ল কালাৰটো হৈছে mosaic endosperm?
Yes, ma'am.
Okay. So this was all about endosperm, okay? From this part, just study the examples. Next heading kora, the second stage or the second event, that is development of embryo. Heading kora, development of embryo. So who forms the embryo? It is the zygote that forms the embryo, right? Zygote কেনেকে form করে? It's very well understood. Very simple question. During sexual reproduction, both the two gametes, the male gamete and the female gamete, fuse together to form a cell that is called a zygote. The zygote ultimately undergoes cell division and forms the embryo, right? This study of embryo is called as what? It is called as embryogeny, right? So here are the basic point কেইটামান লিখো। Right? So what is embryogeny? The study of embryo. লিখি লোৱা। Or the development of embryo. From the zygote is called as embryogeny. Next, whenever we talk about the embryo to co-develop করে। It develops at the co-develop কৰিব? Embryo co-develop কৰিব। inside the ovule, obviously, and you have to be more specific as to which side of the ovule. Whenever we talk about the ovule to dubhagot jodi bhag koru, we are talking about the micropilar end and the collageal end. So embryo formation in most of the cases ko dekha pa kuntu endor. Develop
Micropilar.
at the micropilar end. Very good. Develop at the micropilar end of the embryo sac. The next point about embryogeny is in most of the cases, okay, I think you remember whenever we are talking about fertilization in case of angiosperms, we often use a word that is called as a double fertilization, right? And whenever we talk about double fertilization, দুটা event হৈ আছে সেইকাৰণে তাক আমি কি বুলি কওঁ double fertilization. And what are the two events we are talking about? The development of the zygote and the formation of the embryo and the formation of endosperm. So embryo sac-ৰ ভিতৰত দুটা event হৈ আছে, endosperm হৈ আছে আৰু embryo form কৰিছে. And now the point is দুইটা event একেলগে হয় নে এটা event অকমান আগতে হয়? Does the two events take or occur or develop simultaneously at the same time, or we can see the development of one structure prior to the other structure? কি হ'ব কোৱাচোন? The answer lies in if you know what is the work of endosperm. Endosperm কি হয়? Tissues, right? And what is the most important function of these tissues? Endosperm is what's important? Yeah, it's to provide nutrition. Provide nutrition to whom?
Embryo.
To the embryo. So ultimately, when we are talking about an embryo sac, in majority of the cases, what we will see, embryo development occurring first, endosperm development occurring first.
Endosperm development.
Endosperm development.
It is always the endosperm development. Obviously, okay, that two exceptions are exceptions to so what is it? But in most of the cases, we see endosperm formation earlier or prior to the embryonic embryo formation, and the reason behind that is and growing embryo nutrition or nutrition kini কৰ পৰা পাব endosperm tissues of পৰা পাব, right?
Yes, ma'am.
So সেইটোৱে লিখা in most of the cases the zygote divide only after certain amount of endosperm in most of the cases the zygote divide only after certain amount of endosperm is formed. And reason to just bracket or likhi do that to just in few words sentence of nilikhu kiho karone most of the cases zygote divides only after certain amount of endosperm is formed. Kiho karone to obtain nutrition from the endosperm. Right? So, এইটা আসলে basic point about embryogeny. What is embryogeny? The development process. Zygote of embryo হোৱা process টোক আমি কি বুলি কয় embryogeny. It develops at the micropilar end. It develops at the micropilar end of the embryo sac, and in most of the cases, endosperm to agote form করে, after zygote form করে. Now, and the next point is, whenever we talk about embryogeny, we have to talk about two types. That is embryogeny in case of dicots and that too, also in case of monocots. The point to remember this: initial stages of embryo development of a dicot and the monocot are almost the same. Right? প্ৰথম few stages ডাইকটত কেনেকে এমব্ৰায়টো ফৰ্ম কৰে আৰু মনোকটত কেনেকে এমব্ৰায় ফৰ্ম কৰে আৰম্ভণি যোনখিনি stage these are almost similar, because we know obviously they had to come from the zygote. Zygote is a product of sexual reproduction. Zygote to cell division koribo lagibo, embryo to form koribo lagibo, etc., etc. Right? So now we are going to talk about both of them. First, heading koriba, development of dicot embryo. Ekini over, sir? Is the last point over? One hai? Likhilwa. If it's over, please tell me.
Yes, ma'am.
All good? Okay, heading for development in dicot embryo, the development of dicot embryo. That likha, most common type of dicot embryo, most common type of dicot embryo is studied in. So dicot embryo boli kole kiman bilak, there are so many dicots, right? Dicots are kiman bilak plants ase. Kintu most commonly studied embryo kuntu, that is something that we are going to write. So the most common type of dicot embryo studied is has been studied in a plant called as shepherd's purse. Shepherd's purse কথা আগতে কৈছিলো নেকি? Scientific name নাই কোৱা ন? Capsula bursa pastoris. Capsula bursa pastoris family Luciferi. Right? And this type of embryo is called as crucifer type or onagra type of embryo development. Right? So we are going to study the development of the embryo in case of a dicot plant, and which plant has been taken into consideration is shepherd's purse or Capsula bursa pastoris. Now, talking about the embryo development in case of dicots, no, depending on plants to plants, we see certain different types and the different stages also, right? Protectal plant of the embryo development exactly eké hobo, not necessary. So accordingly, we have different types of embryo within the dicots, but within your syllabus, that is onagrad. Tar upori beleg cancer embryo ami beleg type of ase, that is something that you are going to do with your higher studies, right? Now, as of now, we are concerned with only one type. So Dicot embryo boli kole, we are concerned with the onagrad or the crucifer type. Why is it called as crucifer? Obviously, because it belongs to the family Cruciferae. The plant taken into study was Capsula bursa pastores, or commonly known as the shepherd's purse. এখানে লিখি লোৱা. This is just the introduction. Next is we have to see যে এটা জাইগোটৰ পৰা সেই entire embryo to embryo পৰা plumel তাৰ পৰা radical ultimately ইমানখিনি development কেনেকৈ হয়. Obviously it starts from a single cell and the cell needs to get divided and redivided to form all those structures, right? So এইখিনি লিখাৰ পিছত the next part is something that we are going to study in the form of a diagram and not in sentences. আমি এই বস্তুটো লিখি নাযাওঁ that the zygote undergoes cell division, it forms two cells, etc., etc. At a diagram akim, the diagram is going to speak for it and just see whether you will understand or not. Our diagram akibole okon dangor ke page ekhon loba because it's quite a big diagram. Once you are over with this, let me know. Is it done?
Yes, ma'am.
মুছিব পাৰো গোটেইখিনি?
Yes, ma'am.
Okay. So এতিয়া will draw a diagram. তোমালোকেও okay. So figure to কি হ'ব? প্ৰথমতে figure to লিখিয়ে লৈছো.
মেম এটা স্ক্ৰিনৰ বাহিৰত গুচি গৈছে কি লিখিছে আপুনি
Screen figure development of dicot embryo screen বাহিৰত গৈছে. কিমানখিনি
Yes,
গৈছে
ma'am.
বা? The entire went away or what?
No, ma'am.
ডাইকট এমব্ৰায়টো লিখিছে
Is it visible now? Forget it. Doesn't matter. Likha figure development of dicot embryo. It is just a figure that we are going to draw. Namdo. Okay. Since we are talking about the embryo development, embryo kuntu cell of development, it starts developing from the zygote. So the first diagram that we are going to do is obviously the cell called as zygote. The next point is whenever you go for this type of a diagram, right, you have to take into consideration the ovule also. You have to pay attention to the two words, micropyle and cellagia. I mean, zygote junto side thakibo, that side obviously would be the micropyle. Right? So if I am talking about the diagram, suppose this is the zygote. Can you see this? It could দেখা পাইছা নাই? Diagram to?
yes ma'am
Next screen पर उला গৈছে নাই উলা no? Okay.
পাইছো
Okay. So zygote to বুলি কোৱা লগে লগে এইটো সাইড কি হ'ব? Micropylar উপৰ পিনে কি হ'ব? So zygote to ক'ত develop কৰিছে? If you're talking about this one as the embryo sac and so zygote ক'ত develop কৰিছে? ইয়াত এইটো কি হয়? Micropyle, এইটো কি হয়? Zygote. We are drawing which type of a ovule? We are talking about an anatropous type of a ovule. Why anatropous? Because this is the most common type that is seen in case of angiosperms. Right? Can you see this part reflection has any ke?
No, ma'am.
No, ma'am.
Hmm.
मैम हेतु हॉस्टोरियल की लिखा আছে मैम
Hostorial cell.
Suspensor number to exactly 5 হ'ব নে নে বেছি কম হ'ব পাৰে?
No, they are around about 6 to 10. So any number.
আমি এইটো hostorial cell লিখা আছে ন?
Huh. Is this part of the board visible? A PowerPoint যোনখিনি লিখি আছো.
no ma'am
It's not visible?
নাই।
এইটো part?
No, ma'am.
Okay. Can you see the diagram?
মেম
yes ma'am
Diagram দেখিছা no? Okay, let's do one thing. তোমালোকে নুমুচিবা. Is this part visible?
Yes, ma'am.
This part?
Visible.
yes ma'am now it is visible
Okay. Okay. প্রথমে লিখিলো and then we'll understand.
Bye.
Hello.
Just we are going to see that embryo to কেনেকে form কৰিছে zygote এটা cellৰ পৰা, right? Can you see the diagram clearly? Yes or no?
Yes, ma'am.
Yes, ma'am.
এইটো পাৰ্টত লিখিলে দেখা পাবানে? The portion that I am drawing, just see. Can you see? এই যে arrowhead গিডাল দিছো.
Yes, ma'am.
Can you see this word?
Yes, ma'am.
So this is the first step. Is the green color visible on the board?
Yes, ma'am.
So this is the second diagram. From here, it is this one.
मैम नीचे में क्या लिखे हैं
कौन सा?
इस तरफ लेफ्ट साइड में
यहाँ पे बोलो. यहाँ पे.
नहीं नहीं मैम
यहाँ
इस
पे?
तरफ इसमें लेफ्ट साइड हाँ ये लास्ट वाला
ये
लेफ्ट
वाला?
साइड लेफ्ट साइड
लेफ्ट साइड.
लास्ट डायग्राम
ये
जो
वाला.
हाँ हाँ हाँ
हाँ
हाँ
इसमें. इसमें कौन सा? ये वाला.
मैम वही हाँ मैम
ये वाला ना?
हाँ
हाइपोकोटाइल. HYPOCOTYL. हाइपोकोटाइल. ओ थोड़ा सा कट गया ना वो. क्या हो रहा है? Is the word visible now?
Yes, ma'am.
Okay, complete the diagram and then we'll explain. Overall, look for diba. Is the diagram over?
Yes, ma'am.
Okay, so let's explain now. Okay, see, we are talking about the development of the embryo. Who forms the embryo? Obviously, the zygote. Zygote to basically, when we are talking about the ovule, it is towards the micropylar end, right? So we have the zygote. Zygote is one cell that ploidy kiman twice in. So this zygote, if it has to form an embryo, right? So obviously it has to undergo cell division. So at the first step, what we see is this cell got divided into two cells, terminal cell and the basal cell, right? This terminal cell is also called as embryonal cell, and this cell divides in all plants to form a pro-embryo, right? So it divide and redivide peripele as a collection of cells form koribo. Kiman cells as at the first stage, that is 4 cells. Right. One thing that you have to pay attention is the word called as pro-embryo. What we are studying is the development of embryo. Up till here, in any of the diagram, can you see the word embryo, or you are just saying some words called as pro-embryo? Embryo word to dekha paisa jano. Can you see that word? Okol embryo likha ase niki kobat?
No, ma'am.
Right? No, what is the topic of discussion? Development of the embryo. So even up till here, we cannot say that the embryo is formed. For those students who have studied, or maybe you know the difference between the pro-embryo and the embryo, and or maybe when can a pro-embryo called as an embryo? We'll come to that later. As of now, So the first structure that is formed is proembryo. This proembryo is of 4 cells. Similarly, this basal cell will also divide and re-divide to form a long line of cells that is called as the suspensor. What is the function of the suspensor? A little later. The 2 cells, this cell divide and re-divide to form a round structure that is called as proembryo. This cell divided and form a linear structure that is called as a suspensor. Suspensor to generally kiman thake 6 to 10 cells thake. Right. Now, the last cell of this suspensor is called as the hostorial cell. And whenever we use the word hostorial cell, obviously what we understand is the function of this cell is to absorb the nutrients. It acts as a hostoria, right? So it will absorb the nutrients and ultimately develop to a development process of help. So this is the long suspensor. The next step is these 4 cells of the embryo will again divide to form human cell structure. Sarita, eta keta form korile 8ta. So this one or this 4 cells divided again to form an 8 cell structure, and this 8 cell structure is called as octant or pro-embryo octant. Right. Next, what about the suspensor? The first cell of the suspensor or the cell that is attached to the pro-embryo enlarges, and now this has a specific name and it is called as hypophysis. So what is hypophysis? The cell of the suspensor that attaches to the octant that is comparatively bigger than the remaining suspensor. Clear? Yes or no?
Yes, ma'am.
Okay. Next is what next? This 8-celled structure or this octant will undergo a type of division that is called as division. Periclinal division. What is periclinal division? Anticlinal division or periclinal division. There are 2 types of division. When we are talking about cell division, 90 degrees that you divide for it is anticlinal. If it divides parallelly with the cell wall, then it is called as periclinal. So if you are talking about a cell, so it was a cell, right? Parallelly divide koribo labo. Keneke? It has a cell one hoy. If this is a cell, right? So if this is a cell, this was one cell. If it has to divide in a periclinal way, it will parallelly NK divide koribo labo. So it will cut off 2 cells. Is the point clear?
Yes, ma'am.
Right? So ultimately, this 8-celled proembryo divides periclinally and ultimately it forms a 16-celled proembryo. And what about this hypophysis? This hypophysis also divide and re-divide to form a collection of how many cells? 8 cells, right? So we have a structure, yate keta ase 16 cells, ekinit keta ase, and this is the suspensor that will degenerate later. As of now, we are concerned with all the steps of this structure. If you see, yate keta ase 16 cells, the first 8 cells, no? It is the imaginary, a line buli dhoru. The first part of this structure will form ultimately the plumel and the cotyledon. The later set or these 8 cells will form the hypocotyle. Talking about the hypophysis also, now the hypophysis will be 8 cells. Out of these 8 cells, the first 4 cells will form the root cortex and the last 4 cells will form ultimately the root cap and the root tip. And 8 duita log lagi pelai ki form koribo laibo radical. So now we have an idea about that embryo development. We know from the zygote the embryo will get developed. Our embryo ultimately ki form koribo, plumel, cotyledon, radical form koribo na kore, but baby plants will develop, right? So we have to know which cell is responsible for the formation of which part. Plumel kune form koribo, radical kune form koribo. It is always this part of the 16-cell embryo that is responsible for the formation of the plumel, and it is the hypophysis that is responsible for the formation of the radical. Aru upor part root cortex and lower part root cap. Now coming to a word called as hypocotyle. Have you heard the 2 words called as epicotyle and hypocotyle? Unisa niki? Hypocotyle or epicotyle? right? So it is that part. What is the hypocotyle? It is that part from which the radical will develop. So ultimately, ekhini portion of ki hobo radical will develop koribo, right? So hypocotyle ko ki a line dalo upor khini plumal, major khini hypocotyle, or tarpisot radical. So hypocotyle is that area from which the radical will develop. What is epicotyle? We'll discuss it little later. Ekhini diagram clear hoise na nai?
Yes, ma'am.
Yes.
Yes, ma'am.
Okay. Now we are still using the word called as pro-embryo, but we have to talk about embryo formation. So we need to talk about the pro-embryo embryo form keneke korile, right? So we know you have to write a line, and that will explain. At the beginning, the embryo is globular in structure. This globular structure changes to a heart-shaped structure, and this heart-shaped structure finally becomes a horseshoe-shaped structure. আর যেতিয়াই a horseshoe-shaped structure হবো, then it is called embryo. Just read the line whether you have understood or not. So we are using two words, one is pro-embryo and embryo. So if you have to use the word embryo, then obviously it should have a specific shape. And what is the shape that we are talking about? Horseshoe. So in case of angiosperms, embryo development we see certain changes in the formation of the embryo. Embryo question what are the steps of transition in forming the dicot embryo? So you have to remember globular to heart shape to horseshoe. Jetia horseshoe shape finally takes the structure, it is only at that time you can use the word embryo. Prothomote ami ki use kori asilo pro embryo. Why? Because it is not a horseshoe shaped structure, instead it was a globular or a spherical structure. A spherical structure to ki hobo laibo heart shaped hobo laibo, heart shaped to bisat ki hobo laibo horseshoe, and tetyahe ami embryo word to use koribo pari. Is the point clear? This is very important, so you need to remember these changes. Globular to heart shaped to horseshoe shape. So angiospermic embryo shape kenekwa horseshoe. Clear?
Yes.
Okay. Can I rub this part? Ekhini musu?
Yes, ma'am.
Okay. Now come back to this diagram. Etu ki hoy ase globular hoy ase ki form koribo laibo. So this is ultimately a globular structure, right? So if you are talking about this one, something like this, eneke globular structure form kori ase. A globular structure to ki hobo lagibo? Heart shaped hobo laibo. Heart shaped hobo lagile. A round, if you are talking about this as a round structure, if it has to get converted into a heart shape, a structure ki bahi jabo lagibo ni na lage? Are you understanding? Right? So ultimately, from the globular structure, what we see is a structure like this, right? The plumule will arise. We'll talk about it later, right? Next, yate ki asile that. So if you look at this diagram, A part cotyledon. So this was a globular structure. This was the line. Maybe we are talking about this line, right? So key form they will divide A2 portion or A2 portion of a basic cell division. So they will form a raised structure and ultimately they will take a shape of a heart, right? Our baki cells will have to if you're talking about this, to hypocotyle acili, right? And then this part may be the radical. Are you understanding? Rough diagram. Right? So ekini part of ami ki boli koisu? Hypocotyle, and this part radical. So this is the structure, right? This part was the hypocotyle, this area. This is the area that we are drawing, the hypocotyle area. So key form korbo, the radical, the root tip, the root cortex, etc. Right? আৰু ইয়াৰে ওপৰত যোনটো এই 8টা cell আছিলে তাৰ ভিতৰত কি হ'ব that this part and this part will elongate and ultimately it will form a horseshoe. So exactly what it forms is it will form the cotyledon, and since we are talking about a dicot, keita cotyledon form koribo? Duta and obviously the plumule. Is the point understood? Globular structure transformed into key heart-shaped structure. Heart-shaped keneke hol the yar basic cell division hole and then ultimately form the heart-shaped structure. Center of develop plumule to develop plumule. This raised structure developed into the two cotyledons of dicot. We have the hypocotyle. From the hypocotyle, the radical got developed. We are talking about the transition from pro-embryo to embryo. From this heart shape, key form right? Horseshoe shape form embryo. What happens is when we are talking about this one, this area, no, A2 area, cell division so cell division, this will keep on increasing and increasing, and after some time, what happens is this portion will bend. A portion to cells, once it goes up, after some time, it no longer goes up. Instead, it will bend. This cotyledon also, it will go up and ultimately it will bend over the other cotyledon. So the diagram that we have is, so we are having this, maybe the hypocotyle, we are having this one as the radical, right? A portion to keyhole, this was the plumule, right? What happened to the cotyledon? The cotyledon bent the first cotyledon. Like keyhole, bend hole. Secondly, the second cotyledon also bended over the first one. Is the point clear? বুঝি পালো নে?
Yes, ma'am.
Right? So, cotyledon কি হৈ গল? Bend হৈ গল. And this shape is referred to as a horseshoe, and it is only at this time that you can use the word embryo. Is the point clear? We had a globular structure. From the globular structure, right? Cell division started in this portion, and ultimately the cell divided to form two cotyledons. After a certain time of cell division, the two cotyledons bended one over the other, and ultimately it's taken a horseshoe-shaped structure. And at this time, we can use this structure, or we can call the structure as an embryo. So this is the way in which the embryo is formed in case of a dicot plant. It starts from the zygote. Zygote of কি হয় 4-cell pro-embryo. From 4-cell pro-embryo, it turns into 8 cells, from 8 cell to a 16-cell structure. Out of the 16 cells, 8 cells are responsible for the formation of the two cotyledons and the plumule. The remaining 8 cells are responsible to form the hypoprotide, which is that area from which the radicle will develop. Who is responsible for formation of the radicle? It is the hypophysis. What is the hypophysis? The cell of the suspensor that remains attaches with the octant, right? That is the hypophysis. So is the point clear? So akiba part buzi poa nai niki? Is it clear?
Yes, ma'am.
Right? So এই stage কেইটা মনত ৰাখিবা যে globular পৰা কি হল ultimately horseshoe shape হোৱাৰ পিছতহে we can use the word embryo. Any questions from your part? Can I rub the board now?
Yes, ma'am.
So this is how the embryo develops in case of a dicot plant, right? Now, if you want to see the embryo, no dicot plant or dicot embryo to see sabobisara, where can you see it? Obviously, inside the dicot seed, yes or no? Dicot embryo ko bhitor dekha paba, dicot seed er bhitor dekha paba. So, if we are talking about the seed, so dicot seed or jodi structure ta ako, then we draw something like this, a rough diagram. ইয়াতে কেইটামান টাৰ্ম আছে এই টাৰ্মছ কেইটা জানিব লাগিব Is this green line visible?
Yes, ma'am.
So this is the rough diagram of a dicot seed. So obviously, you have certain terms called as cotyledonary, epicotyle, hypocotyle, radical, right? So what is hypocotyle? What is epicotyle? First, this one called as cotyledonary node. The two cotyledons meet at an area, and this meeting point is called as cotyledonary node. The portion that is above the cotyledonary node is called as epicotyle. Epi mane ki above the cotyledonary node. This epicotyle ultimately give rise to plumule, right? Similarly, the portion that is below the cotyledonary node is called as what? Hypocotyle, and it will give rise to radical. Next, we have an imaginary axis. This axis is called as embryonal axis. Tigulum word to monotractable. So, what is an embryonal axis? The hypothetical axis. So, talking about the dichotomy, these are the structures that you are going to see. Definition ahibo pare for your both epicotyle ki, hypocotyle ki, etc. etc. So, figure dicot seed. Is it clear? So what we have discussed is we have talked about the embryo development, dicot embryo we have talked about, and where can you see the dicot embryo? Inside the dicot seed. So what is the structure of a dicot seed? This is all that we have discussed. After this, we need to move on to the development of a monocot embryo. What we said is when we talk about the development of embryo, either it is dicot or monocot, initial stages are the same. So once you complete this, please tell me so that I can move to the monocot part. Complete হৈছে নেকি?
Yes, ma'am.
Yes, ma'am.
Okay. Ready for the development of monocot embryo. Right? আকৌ সেই তেনেকে introduction অকমান লিখি লওঁ. The common type studied is the embryo of a plant called as Luzula foscaris. This type is called Sagittaria type of embryo development. In case of Dicot, we have used the word called as Onagraph type or crucifer type. Now we are using a word called as Sagittaria type. In such a type, so these are the first 2 points. Next, again, diagrammatic study.
Ma'am, Sagittaria কি লেখা আছে?
Type of embryo development. Sagittaria, Sagittaria type of embryo development. So since we are talking about embryo, obviously we'll start it from zygote. Zygote ki hoy, twice in. Talking about the embryo, we have to consider the micropylar area and the collagen area of the ovule, right? So yate ki hoy, aako diagram. Ito iman ta complicated diagram nohoy. In this case, if you see the diagram, automated duta cell, now we have a collection of 3 cells. The basal cell does not divide. It is the terminal cell that will divide and form 2 cells, that is the terminal cell and the middle cell. So we have a structure like this. Let's draw it here. So we have a structure like this. It's a terminal cell box, it's a middle cell box, and it's a basal cell box. Right. This terminal cell will ultimately form is this part of the board visible? Something in the bracket. দেখা পাবা নে? Is it visible?
Yes,
Yes,
ma'am.
ma'am.
Out of the 3 cells, 1 cell will form the corpuscle, the other cell will form the embryo. This cell will divide and redivide and form embryo. A basal cell, as I said, it will degenerate later. So after this diagram, we'll see this diagram. Right? And then, so again in this diagram, what we have said is, we have started it from the zygote. The zygote divides into two cells, the basal cell, terminal cell. The basal cell will not divide, terminal cell divides into two. And ultimately, the terminal cell will form the cotyledon, the middle cell will form the embryo, and the basal cell will degenerate. So we are concerned with just these two cells. No? Out of this one will form the cotyledon. And as we have discussed in case of dicots, no, cotyledon will ki hobo, since it starts forming, no, form kori cell division whole play, when it forms a huge amount, it starts bending. So in case of monocot also, we will see the cotyledon in the form of a band. Dicot jenike straight ho bela bend ho gosle. Monocot to ki hobo, it will bend over. So the diagram that we will see is so we're talking about this as the embryo, and this part shall give rise to the baby plant. We will see the embryo here and cotyledons. And what we know about the cotyledon in case of monocot, that it is thin and papery. We see reserved food almost absent. Suspensor cell দেখা পাবো পাৰো নাপাবও পাৰো. And this is the basal cell যোনটো দেখি degenerate হৈ যাব অলপ পিছত. So this one is the cotyledon. Right? From this diagram, this is the embryo. A embryo to prakiubo, enake pleumal to develop korbo, enake radical to develop korbo, and this one will bend down. Is this diagram clear? Etu diagram or pisot kuntu diagram? This one. Is it clear? Etu keneke enuka structure form korile dhoribo parisa ne nai?
Yes, ma'am.
Right? So if this is the embryo, please look into the book. If this is the embryo, ekhine jodi embryo form kore, embryo pra ki form koribo? A baby plant dal form koribo above and the part below, right? So this will form the pleumal and the radical, and the embryo will bend over. So this, sorry, and the cotyledon will bend over. We have seen the cotyledon in case of monocot. The next point that you need to consider is, we do not use the word pleumal and radical generally in case of monocots, right? Instead of that, we use two other terms that is called as coleoptile and coleorhiza. Ekita word shunisan na agote? Coleoptile, coleorhiza. What is coleoptile and what is coleorhiza? Ki hoy? What about the rest of the students? Have you heard these two words?
Yes,
Yes,
ma'am.
ma'am.
Yes or no?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Ki hoy?
I'm covering.
Yes, ma'am. Covering of
Covering.
pleural and
Covering of?
radical.
Covering
Pleural
of
and
pleumal and radical. Actually, the best way to answer is coleoptile is nothing but the plumel. Coleorhiza is the radicle. Now the point is, when will you call a plumel a coleoptile and the radicle a coleorhiza? When the plumel is covered by sheets of cells or layers of cells, and the radicle is also covered with sheets of cells. So in this case, or in case of monocot, আমি কি দেখা পাওঁ? Plumel radicle to ওপৰত কি থাকে কিছুমান layers থাকে, and ultimately this part is called as coleoptile. So the question is asked as the coleoptile and coleorhiza are the is the characteristic of which group of plants? It's the characteristic of monocots and not of dicots. Dicots of কি দেখা পাওঁ? Root, sorry, plumel or radicle. ইয়াতে কি দেখা পাওঁ? Coleoptile and coleorhiza. Right. Next, if you're talking about this cotyledon, right? Cotyledon, in case of so this cotyledon has a specific name, no?
Yes, ma'am.
Monocot or cotyledon to কি বুলি কওঁ?
Scutellum.
Very good. It's also called as scutellum. And this scutellum is thin and papery. It has, it information লিখিলোৱা, has negligible food reserve, almost nil, no? And this figure ki development or is this clear? একেলগে কিবা questions? Right? Next is, if you want to, again, as we have discussed it during a dicot embryo discussion, if you want to see the embryo, you have obviously see the dicot seed. Similarly, in case of a monocot embryo also, monocot embryo to sabo lagile, we have to talk about the monocot seed. So I think this is all for today. In the next class, we are going to talk about the structure of the monocot seed. Right? So আজি এখিনিলৈকে থাকো। এইটো portion লৈকে কিবা questions আছে নেকি?
Ma'am, scutellum খালি monocotor কাৰণে কয়।
Oh, the single cotyledon in case of monocots is called as a scutellum.
Okay, ma'am.
Here? Okay.
Ma'am. Ma'am, coleoptile কিহক মানে কিহলে convert হয়?
Plumel, the shoot portion.
Okay.
Coleorhiza will give you the radicle or the root portion.
Okay, ma'am.
Okay. Any more questions?
Ma'am. Ma'am, coleoptile and coleorhiza আপুনি covering টোৰ কথাটো কোৱা নাই না মেম, পুৰা পাৰ্টটো কথা কৈছে।
পুৰা পাৰ্টটো coleorhiza অকল covering নহয়, যোনটো portion of shoot ওলাব ন বা plumel খিনি ওলাব ন, সেই portionটোত আমি plumel বুলি কেতিয়া কম যেতিয়া সেই covering খন নাথাকে। যদি covering থাকে ন, extra layers of cells থাকে, তেতিয়াহলেতো আৰু চোৱা dicotত নাথাকে, monocotত থাকে, you cannot use the same term in both the cases. So in one case we are using the word plumel, in the other case, if that portion is covered with sheets of cells, we use the word coleoptile and coleorhiza. Ultimately, plumeলৰ পৰা কি হ'ব? shoot to arise কৰিব, coleoptile পৰা কি shoot portion to arise কৰিব। Just the two terms that is used in case of monocots. Clear?
Yes, ma'am.
Yes, ma'am.
Any more questions? Okay then. Thank you for today. We'll meet on the next class. For the next class, heading for the structure of a monocot seed. Thank you.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Welcome.
हमारे शेयर